as we will see in the next section of the course, when we move from organismal and organ system aging to cellular and molecular aging, deterioration and functional breakdown, in other words, aging, often begins at the level of cells and molecules. Since in this section we're talking about organismal aging, let's discuss one of the most important organ systems that is involved in the aging process, the brain. Many scientists believe that the brain and neuronal systems in general are particularly susceptible to the effects of aging. For the longest time, scientific consensus has held that the brain, once fully matured, was largely immutable and did not change cell numbers very much. Thus, no new neurons would be produced over a full lifetime, and any loss of neurons would be irreplaceable. This was in stark contrast to other organs and tissues, which contain so-called stem cells. Stem cells are cell populations that are able to reproduce indefinitely and thus are able to repopulate organs and tissues that experience cell loss. We will talk about stem cells in a later chapter. But based on these considerations, it is only natural to conclude that a brain that loses cells as a consequence of age-dependent decline cannot replace them, and thus is more susceptible to aging. In turn, as the brain is the control organ for the rest of the body, brain aging has outsized effects on all other organ systems. This view of the brain has somewhat changed over the years, especially with the discovery in the early 90s of adult neuronal stem cells, or ANSC. In subsequent experiments, these cell populations were found to be able to create new cells for all brain tissues and replace lost cells as a consequence of damage, disease, or even aging. Many researchers further think that these stem cells are also responsible for the remarkable brain plasticity that is often observed in damaged brains, for example after a stroke or after an accident, like in this case with a railroad rod right through the brain. These adult neuronal stem cells are, however, very slowly dividing and are very rare. Two regions in the brain where they have been found are within the hippocampus, and two subzones of the hippocampus called the subgranular zone, SGZ, or the subventricular zone, SVZ. It is currently an area of very active research to find more regions of adult neurogenesis and more and different ANSC populations. In fact, a paper just came out early in 2018 that questions whether adult neuronal stem cells can exist in the first place. While these findings certainly improve the dire outlook on our brain, and that not all is set in stone that the brain can recover and can make potentially new stem cells, they still highlight the fact that the brain is indeed fairly susceptible to cell loss, and thus very susceptible to the aging process itself. As we have seen before, organismal aging is highly complex and varies from organ system to organ system. One problem with the brain as an aging organ is that it itself is highly varied, with many extremely specialized areas. For example, there is the hippocampus, the area we just talked about, the region where potentially adult neuronal stem cells were discovered. The striatum, a region of motor neurons that controls all the motor function of the body. Other than being made from similar cells, such as neurons, astrocytes, or oligodendrocytes, these regions have very little in common, and in fact respond to aging very differently. The investigative approaches to brain aging similarly need to be very diverse. Since, once more, we can't really experiment on humans, researchers can use behavioral approaches to test functional brain decline by testing memory, for example episodic and semantic memory, perception and visuospatial ability, and stimulus processing and decision-making called executive function. Performing different tests for different functions thus allows for testing of different areas of the brain and give important insight into pathologies of the brain. It thus allows one to pinpoint specific damages. This is particularly important when it comes to one of the most commonly associated problems with aging, dementia. Dementia simply refers to a loss of cognitive function that is a decline from a person's previous intellectual state. but does not always include amnesia, which is memory loss. Moreover, dementia, or its preclinical state, mild cognitive impairment, or MCI, is not required for aging. It is rather a sign of some underlying disease pathology instead of aging per se. These syndromes are a collection of signs and symptoms, are indeed more prevalent in the elderly, which is why they're considered age-related diseases. But it bears repeating that they're not binding as a consequence of aging. While the behavioral tests can reveal functional decline of an aging brain and give hints to which area of the brain is most severely affected, they provide no insights into the causes and mechanisms. This is where modern technology comes in, especially in the form of MRI and PET scans, which enable peeking into the skull and visualizing a living brain. The biotechnology revolution also brought us the ability to analyze the biomolecules of a living brain, for example, by analyzing the specific proteins in the cerebrospinal fluid. It also allows us to perform genomic sequencing to detect gene variants in the genome of a person that are associated with brain decline. Expression profiling to detect which genes are turned on and off in a brain or section of a brain. Often, incorrectly expressed genes are contributing factors in functional decline. Or proteomics, the analysis of all active proteins in a sample to detect dysfunctional and damaging protein variants. Magnetic resonance imaging, or MRI, is based on a phenomenon called NMR, or nuclear magnetic resonance. It relies on the fact that atomic nuclei are able to absorb electromagnetic radiation, such as radio waves, and re-emit them. Individual elements, and even isotopes of elements, have very specific behavior when exposed to these electromagnetic fields. In practice, this means that powerful magnets are positioned around the brain to be scanned, and hydrogen nuclei excitation is performed at various field strength. Since the specific microenvironment of a particular nucleus can affect its behavior in the electromagnetic field, this means that changes in brain structure can be visualized and become apparent as discolorations on an MRI image. For example, in multiple sclerosis, neurons lose the myelin insulation of their axons and consequently die, leading to lesions in the brain. In an MRI scan, these lesions show up as bright spots due to their slightly altered NMR behavior. Lesions in patients with age-related diseases can thus similarly be detected. PET scans, or position emission topography, is a technique that uses radioactively labeled biomolecules as they pass through the body and become metabolized. In a typical PET experiment, this radioactive tracer is visualized through a series of X-ray images, which are then assembled and processed into a 3D image. Radioactive tracer molecules could be analogs of glucose, for example, which gets preferentially metabolized by active brain regions. Similar to an MRI scan, a PET scan can thus detect brain regions that inhibit functional decline. Increasingly, molecular approaches are being used in evaluating brain function. This includes investigating the protein content of the cerebrospinal fluid, which is the liquid that the brain is suspended in. The CSF has a very different molecular composition than regular blood due to the creation of the blood-brain barrier, extremely tightly linked cells that line the blood vessels that support the brain. The BBB is only permeable for very select molecules, such as certain lipids or molecules for which specific transporter molecules exist. As such, the CSF is lower in protein content, meaning that a slightly altered CSF composition may hint at brain abnormalities. Another modern approach is to use large data sets, or omics, such as genomics, proteomics, metabolomics, and so on and so on. These large-scale approaches don't just look at a single gene, genomics, a single transcript, transcriptomics, a single gene product, proteomics, or metabolites, metabolomics, but take a wholesale approach. They look at all genes, all gene transcripts, all metabolites, etc., to paint a holistic, all-inclusive picture of brain function. What this really means is often another question, as omics approaches often produce an overwhelming amount of data that is very difficult to analyze and to wade through. What then, taking all these approaches into consideration, happens in an aging brain? Not surprisingly, as the brain contains a multitude of parts with different functions, just as in the rest of the body, these parts are affected by aging differentially, leading to considerable heterogeneity in brain aging. What is clear is that, on average, cognitive function declines, which is accompanied by some degree of atrophy or shrinkage. The older an individual, the worse those effects. However, there is huge variability between individuals, with some declining rapidly, while others maintaining for quite some time. This heterogeneity is not only observed between individuals, but is also apparent in differential decline of specific functions. Some cognitive abilities, such as language function or visual processing, decline very slowly with age, while others, especially memory function, decline more rapidly. Within these categories, individuals who show poor performance in one area often also do poorly in other areas. But here again, this is not absolutely invariable. This figure clearly demonstrates this heterogeneity, both on an individual and a functional level. These observations suggest that single, simplified explanations cannot account for the variability seen in brain aging phenotypes. Another difficulty in determining causes and phenotypes of brain aging is the differentiation to disease phenotypes, especially Alzheimer's disease. Preclinical Alzheimer's disease, or even mild cognitive impairment, is very difficult to diagnose and may hamper attempts to describe normal brain aging. Dementia, as mentioned earlier, while age-related, is a disease state and not an aging state. One area of the brain in which normal aging has been fairly well described is the hippocampus, named after its seahorse-like shape. Incidentally, the hippocampus is also the location of a lot of Alzheimer's disease pathology, as we will see in the next module, as well as the site of brain cell regeneration. Its major role is in memory function, which may explain why memory declines during aging or AD. Interestingly, during normal aging, the number of cells in the hippocampus does not decline dramatically. In fact, hippocampal neuronal loss is highly predictive of AD or Alzheimer's disease. Instead, the connections between neurons weaken, which in turn diminishes their ability to create electric signals and thus impairs their primary function. Shown here is a typical neuron in dark red with many synapses to other neurons in beige or yellow. Microtubule highways within the axon of a neuron transport vesicles filled with neurotransmitters to the axon terminals. Upon arrival of an electric impulse, which is created at the axon hillock and travels down the axon to the synapse, synaptic vesicles fuse with the synaptic membrane and release neurotransmitters into the synaptic cleft. Receptor molecules contained within the dendrite structures of the postsynaptic membrane detect these neurotransmitters and create a signal that is relayed to the main neuronal cell body, where it is computed and converted into an electric signal, which then travels down the axon, and so on and so on. While neuronal numbers in the hippocampus do not drastically decline, what does decrease is the so-called postsynaptic density, or PSD. This term refers to the number of proteins in or at the postsynaptic membrane, including neurotransmitter receptors. Thus, a decreased number of signaling molecules directly leads to a decline in synaptic strength and function. This decline in synaptic function in turn affects neuronal function, including the process of long-term potentiation, or LPT. LPT is the repeated stimulation of neurons, which strengthens synapses and is absolutely critical for memory formation. Another area of the brain that is highly susceptible to aging is the prefrontal cortex and the basal ganglia. These regions are responsible for working memory, judgment, and attention. Also other behaviors, especially anger. During aging, these regions undergo severe changes, which may result in behavioral changes, such as loss of impulse control, poor judgment, and the inability to remember events in chronological order. There's a reason why scam artists often target the elderly, due to the loss of impulse control and increasingly poor judgment as a consequence of decline in prefrontal cortex and basal ganglia function. The stereotypical get off my lawn granddad is likely situated in these areas. As with the hippocampus, cell loss itself is likely only a minor contributor to the aging phenotype. Instead, synapses are lost and dendrite numbers and length is decreased. Nonetheless, the prefrontal cortex is the brain region with the most consistent age-dependent atrophy, suggesting that cell loss in this region may indeed part of the normal aging process. The source of the cell loss may be the dopaminergic neurons, which are lost at a rate of around 5% per decade. The neurotransmitter dopamine is involved in the brain's reward system, which may explain some of the behavior changes seen in the elderly. Interestingly, dopaminergic neurons are also the neurons lost during Parkinson's disease. Although it bears repeating that, just like Alzheimer's disease, Parkinson's disease is an age-related disease, but not part of normal aging. Finally, a region of the brain that is susceptible to aging is the so-called default mode network, or DMN, a very recently discovered, highly interconnected group of brain areas that are active when the rest of the brain is at rest, for example, doing daydreaming or self-reflection. Interestingly, the DMN accumulates high amounts of A-beta, one of the suggested causative agents of Alzheimer's disease, even in normal, non-AD individuals. The greater this A-beta accumulation, the greater the functional loss displayed by the DMN. Thus, A-beta accumulation appears to be part of normal aging, but it still remains unclear what the functional significance of this is, or why the DMN is particularly susceptible to A-beta.
Check your understanding. Explain why the brain is vulnerable to aging and disease. Describe the behavioral tests used for analyzing aging brains. Describe imaging technologies used in brain studies. Describe molecular approaches to test brain function. Describe the roles of the hippocampus, the prefrontal cortex, and the default mode network during aging.